talk a little bit more about body axis patterning. And up to now, we've been thinking mainly in the fly about the anterior and posterior axis. And so we're going to start out talking about the dorsal ventral axes. And you'll see some of the same mechanisms that are employed, but now we have a different spatial orientation and a different goal for the embryo to accomplish. But it can use in principle some of the same tools to construct this information across the body. And just as an aside, this is a really cool song by Tom Petty, and I wanted to play it for you because it's about learning to fly. It fits really well, but I couldn't get the audio going. So your assignment is to play this sometime today and stare at this photograph. Enjoy. Okay. So, harking back to early oogenesis in the fly and the eggs first being made, we have here in yellow one haploid cell that is the egg cell. Then we have several nurse cells, which are mother's diploid cells, busy creating mRNAs and proteins and shunting them over and dumping them into the oocyte. And we have surrounding this whole thing a layer of cells on the outside called the follicle cells. And the follicle cells also have a really important role to play in patterning that oocyte. So this oocyte, as you are already getting the picture, it's not uniform cytoplasm just from one end to the other, but it has different RNAs and proteins and different kinds of information in different places. So part of what we're going to do here in the first few slides is think about how that actually gets started in the oocyte. And one of the first things that one can notice is that this blue circle, the nucleus, tends to be located initially in early oogenesis, down more at the side that's opposite or furthest away from the nurse cells. And so this will become the future posterior side. And I already mentioned this kind of briefly, but what's happening here is this nucleus is actually synthesizing some RNA here called gherkin, and this will be made into gherkin protein, and they'll see a communication with the overlying follicle cells. And so some back and forth between the oocyte and these follicle cells is going to establish this is the future posterior side. And one of the important things that it begins to do is initiate the formation of these microtubules that we said are running from one end of the oocyte to the other. And they have a certain orientation. You know, microtubules have a directionality to them. There's a plus side and a minus side. And so these two microtubules begin to be initiated or nucleated or synthesized down here following that communication of the nucleus and overlying follicle cells. So then they get put in place in this specific orientation, and as we know, because of that, RNAs like bicoid begin to be localized to the anterior side, and then different molecular motors are carrying some RNAs to the posterior side. Okay. At this point, the nucleus is actually going to move, which is kind of weird, but it physically moves from being located here in the posterior side up to the corner of the oocyte cell, as you can see. And now up there in that corner, that's going to become the future dorsal side to this oocyte and then later embryo after it's fertilized. So it moves up there. And once again, when it gets to that location, it again sends out some more gherkin mRNA and communicates with the follicle cells that are overlying it. And this just shows you some of the experimental evidence that we can use to try to understand the fact that this is happening and when and where. So we have in situ hybridization up here at the top showing you where gherkin mRNA is localized. And you can see it's really discrete. Just right there in the corner of the oocyte and nowhere else, we find a higher concentration of gherkin mRNA, and then it's communicating them with these overlying follicle cells. So here is a double immunohistochemistry where there's been a yellow, fluorescent yellow tag localizing gherkin protein, and then probably a phalloidin tagged moiety that recognizes actin. And so that's just to outline the shape of the cells so we can see them better. So once again, the protein, reflecting where the mRNA was localized, and it's really, really discrete. Okay? I don't think I would have ever predicted this if I just sort of came in cold and said, how would an embryo do this? You know, the idea that you could actually get one mRNA to be so tightly localized into one corner, to me, is kind of surprising. And then here we have more of a chromogenic or a color precipitate method of doing immunohistochemistry, and so instead of using fluorescent marker, you're getting that substrate that changes color into purple. And here are two different views, the longitudinal view and then a cross section right through the embryo at that point. Okay, so the nucleus is moving from the posterior end to this future dorsal side, and here is just a diagram of the different body axes that are beginning to be established here at mid-oogenesis. So keep in mind that each organism now is doing this a little bit differently. In C. elegans, they had a method where the sperm would come in always at one side. The frog, the sperm could fertilize anywhere on that egg, and then you'd get cortical cytoplasm rotation, and that would be the first way that symmetry is broken. And here in Drosophila, even before there's any sperm around whatsoever, we're already setting up some body axes just by asymmetric localization of these maternal factors. Okay. So now let's take this big picture view and start to zoom in and look at what's different about this dorsal side of the embryo versus the ventral side of the embryo. So this rectangle is going to be what's in your next slide, and you're going to see a layer of follicle cells, a portion of the single cell that is the oocyte, and then another layer of follicle cells down on the ventral side. And we'll look at some of the molecular players. Okay? So gherkin, mRNA, and then protein being secreted by the nucleus. It's going to leave, it's going to be externally secreted from the cell and then interact with torpedo receptors on those layer of follicle cells. And as a result of that interaction, we now do not make pipe protein in the dorsal follicle cells. So it's actually, the result is to not make something rather than to make something. Meanwhile, down in the ventral cells, there is no gherkin around, there's no signaling through the torpedo receptors, and as a result of that situation, now we do make pipe protein in the ventral follicle cells. So now we have an asymmetry. Some of the cells are making pipe in the ventral, and they're not making it up in the dorsal part. Okay, then things continue as mid-oogenesis is rolling on. Now down here in the ventral follicle cells, there begins to be this whole signaling cascade with multiple different players. And there's actually a little bit of a space in here between the follicle cells and their plasma membrane and the oocyte and its plasma membrane. And so a lot of these players in this signal transduction pathway are acting actually in that little bit of space in between these two cells. And you do not need to memorize any of these factors. You can see on your term sheet 2 or 3 genes that are the most important from this lecture to know something about. Dorsal is going to be one of them. I think cactus is another. But as a result of this pipe being here, now we go through this whole signaling cascade. And then down here on the ventral side of the oocyte, we have the question of whether or not this dorsal protein will go into the nucleus. And initially the oocyte, of course, just has one nucleus and is far away. But after this thing gets fertilized, now that nucleus goes through multiple rounds of mitoses, and then eventually we'll get cellularization, so we have one layer of cells on the outside of the Drosophila embryo. And about 90 minutes after fertilization, this cellularization portion of the cleavage has been completed, and now we're going to have a layer of cells down here on the ventral side, and dorsal is going to go into the nucleus in those cells because of this pre-setup of having this signaling transduction that was back due to pipe, which is back due to the lack of gherkin signaling. So the timing here is really important. So we get this sequence of events that's happening in the oocyte, then things are sort of paused to let the embryo be fertilized and catch up. And then the final outcome would be is that dorsal goes into the nucleus in the cells there on the ventral side. So this protein named dorsal was actually found through a genetic screen looking for phenotypes that would interrupt the dorsal ventral axis. And in Drosophila, the key thing that you have to keep in mind is that the genes were initially named based on their mutant phenotype. So in this mutant phenotype, what we see is a larvae that is specifically defective in the way it sets up the dorsal ventral axes. And so to assess this, you could look at, different characteristics of embryos as they first start to get a dorsal side versus a ventral side. Or you could also look at molecular markers, say just genes that you know should be expressed in cells on the dorsal side, not so much on the ventral side. And so when they did that to look at these larvae, here's our familiar wild-type larvae with the segments and the bristles and clear areas and so forth. But this dorsalized thing, it looks basically like a blob to us, but the people in the know, this is a dorsalized embryo. So it's overrepresented for dorsal type fates and really missing or underrepresented many of the ventral fates. So that's quite interesting because now we have a genetic handle on some gene that is really key early in development, specifically to set up this axis. And the name of the gene or the name of the protein is dorsal because of this dorsalized phenotype. But then you get to thinking about, well, what does dorsal actually do in the wild type embryo? And what was missing in the dorsalized embryo are the ventral cell fates. So the job of the dorsal protein is actually to provide the ventral fates. So, it gets kind of squirrely here. But I wrote it all out. Okay, gene based on the mutant phenotype, dorsal and cactus. And then here we have these two different situations of what the mutant looks like. And then based on that, you would infer the wild type function, which is to specify ventral fates, or for cactus, that's one that helps specify dorsal fates. And in a biochemical sense, dorsal is acting as a transcription factor. Cactus is a protein that binds to dorsal. So in this decision about whether or not dorsal is going to get to go into the nucleus or not, cactus is a co-protein out in the cytoplasm that's binding to dorsal and basically holding it out there until the right time, 90 minutes after fertilization, until we know whether that signaling cascade from pipe is going to impact the release of dorsal and allowing it to go into the ventral cells on the embryo. Okay, so here is what I just told you. So we have pipe protein in the ventral cells. We get the signaling cascade, and then here you can see a cross section now through the embryo at 90 minutes past fertilization. So we do have a whole layer of cells going around the outside of the embryo. And then the darker green color there, that indicates the presence of dorsal in the nuclei in all those round circles. So the darker green it is, the more dorsal that's present inside those nuclei. And what you can notice is it's a gradient. So remember morphogen gradients? This gets set up for the dorsal protein, where we have an area of the embryo that has a really high concentration of dorsal, and then it tapers off gradually as you get more lateral. And then by the time we made it all up to the future dorsal side, those nuclei have really no dorsal protein in them. So a dorsal is a transcription factor, and it would qualify as a morphogen because of this morphogen gradient and because of different fates associated with different amounts of dorsal protein. So let's at actually an immunohistochemistry to see how this dorsal protein is arranged in wild type and different mutant conditions. So here in the wild type embryo, if we detected dorsal with an antibody, we would see really high concentrations in these ventral cells. It tapers off as you go laterally, and then in the dorsal side there's none. And here is a dorsalized mutant case. So this could be an actual dorsal gene mutant embryo here with no dorsal protein at all. And in that case, the phenotype means that there's no ventral face. This is actually a cactus mutant where we're missing cactus. So in this scenario, the dorsal protein manages to get into the nuclei everywhere. So all the cells around the whole embryo end up having a high concentration of dorsal. The job of dorsal is to provide the ventral fates, and so this becomes a ventralized phenotype with overrepresentation of these ventral fates. Okay, does this make sense? Any questions? Okay, so dorsal acting like a morphogen, and here in the regions where you get the highest amount of dorsal, that's associated with some mesoderm cell fates. And then we have 3 different colors of blue, which indicate all ectoderm fates, but different types, say neurons, a couple different types of ectoderm. Amnioserosa is one of the membranes that surrounds the embryo. Suffice it to say, though, that just different concentrations of dorsal associated with different cell fates. Just as we've seen for other morphogens like bicoid, high concentrations of bicoid was acron, and then lower was more head structures, and lower than that were trunk structures. So it works exactly the same way, only we're using a different gene product, and we're applying it to a different body axis. So we said that dorsal is a transcription factor, and in more of a biochemical sense, what it's actually doing is recognizing different target genes. And high concentrations of bicoid may stimulate high levels of transcription from certain target genes that are needed for mesoderm type cell fates. And so that's how a high concentration of bicoid could end up playing out into differential gene transcription that might be needed by some cells to become mesoderm. And so here are a couple of different gene names. These are some of the targets: twist and snail and rhomboid, and so the targets of dorsal, some of these themselves will be transcription factors, so this sets up a cascade. Some of them are going to be paracrine factors, which you'll recall are short-range signaling molecules, so these might be secreted and travel over to cells that are nearby. Some of them are proteases, so this is an actual enzymatic function that the embryo might need to carry out early in life. Okay, here's another common theme with some of the other transcription factor morphogens we've talked about, like bicoid. Sometimes it has an activating type function, and sometimes it has a repressing function. So the context of all of this is still important. We have here one critical morphogen factor, but that's not the only gene that's in there. And all of these transcription factors are acting together to try to present a unique combination or a unique situation for each individual cell that then responds and differentiates according to the fate that is appropriate for its time and position. Okay. And so each of these different targets would have enhancers, so there are specific binding sites for each of the different transcription factors. So here, this just shows you the readout of a morphogen like dorsal. And these are 4 different target genes. And so we've got here actually an immunohistochemistry that's been probed for 4 different proteins, which are each tagged with a different color. And it may remind you of the gap gene kind of expression. So we get these really discrete bands of expression at different places in the embryo. And this is actually the kind of readout that you could get from a continuous morphogen gradient. You can still get these chunks, domains of expression, because each target has a certain threshold that it responds to for the amount of dorsal protein that's present. So, I was mentioning that a lot of the same mechanisms and themes are involved, and you're starting to see this is true. So here's another little slide that you could use to think about during study time. These are just some of the different concepts or mechanisms that we've talked about as the embryo is faced with trying to establish its body plan. And they all kind of come into play with this body axis too. So morphogen concentration, dorsal is our morphogen. Spatial boundaries, so some of the target genes and how those become expressed in really discrete regions. Temporal means timing, and here we have a certain situation that we need to start really early in the oocyte, and then later on at 90 minutes past fertilization, this plays out into the dorsal going into the nucleus in a certain group of cells. Cross-regulation, so the more genes that are expressed in different timing of development as the minutes tick by, now we get more transcription factors around in the cell to do their own thing and refine the domains of expression and activate or repress even further targets. Translational control, so some of these genes such as bicoid or nanos are not only regulating transcription, whether mRNA is being made, but also translation. So can those mRNAs be translated into proteins? So, this is actually, I have a study guide question that comes back and asks you to see if you can articulate in a sentence what this mechanism is all about, how it works for the embryo, what it does. Okay, any questions on any of this dorsal ventral situation? I'm going to keep going just a little bit here into lecture 13, and then we'll stop in our break, and then we'll have an in-class exercise. Okay, so back when we talked about the segmentation genes, there was one group that we have not yet talked about, and these are the homeotic genes. So that's what we're going to discuss next. And these were first discovered in Drosophila, again as part of a forward genetic screen. This was done by Nusslein Bullhaus and Bieshaus, and they were looking for genes that disrupted larval patterning early on. And so they found a number of those mutant phenotypes that I've showed you over the last few lectures. But another class of mutants that they found was not so much something that deleted portions of embryos, but actually changed the fate of some cells from one fate into a different fate. So it wasn't just that they were broken or missing, but they were altered in what fate they became. And there was actually 8 genes that were discovered that could mutate in this way, and they all turned out to be on chromosome 3. And they're present here in 2 sort of clusters or groupings. So there's a group of about 5, and then there's a little bit of a space, and then a group of 3 more. But nevertheless, these 8 genes are more or less contiguous with each other, so they're linked together on the same chromosome in roughly the same area. Okay? So this first grouping is called the antennapedia complex, the second grouping is called bithorax, and there is actually a gene in here. This is the antennapedia gene, and then here is the ultrabithorax gene. So they're sort of named after one of the genes in the cluster. And overall, this cluster is called the homeotic complex, or the HOM complex. And it's some really interesting features. So here, the adult animal, at the patterning on the anterior posterior axis, specifically at these segments that become the thorax, there's 3 of them. And here's what should happen in the wild type situation. That first segment, that chunk of tissue that we looked at, that should become thorax, and that will give rise to some of that more anterior tissue, including a pair of legs. The second thoracic segment in the larvae will become this back area here, and a pair of wings will be attached and grow out from that second larval segment. The third segment also gives rise to this kind of thorax region, but there'll be a pair of halteres that are connected from that tissue. And halteres are these small little stalks with a round bulbous thing at the end, and the fly uses them to help balance when it's flying around. Okay? So they're easily recognizable structures. These kind of bizarre little appendages hanging off of the fly here. So in the ultrabithorax mutant, which was one of these 8 genes, what happened here is now this third thoracic segment changed its identity, and it now became a duplication of the second thoracic segment. So we have no halteres here because we changed that into something different, but instead we have now a whole second group of wings and a whole second thorax, because that T3 segment got converted into a T2 segment, and then the T2 segment did what all good T2 segments always do, which is make a thorax and a pair of wings. So this is a very different kind of situation, patterning-wise, than just deleting something or breaking it so it's non-functional or kind of mal-patterned or abnormal. It's really it's altering the pattern. And so we have to have a term to describe this sort of situation, and it's called the homeotic transformation. And it means that one body part gets transformed or changed or replaced within the body structure as the pattern develops. Okay, so this is a very special situation that's only possible with a select, unique group of genes. And then we'll have the term homeotic gene, and that we can use to apply to any gene that might mutate to get this sort of specialized phenotype. So it's really, specifying body identity. What is the identity of that segment that would develop later on in the adult, and what are the genes that control that? So if you could mutate those genes and change the body plan this drastically, that gene must be doing something really critical to identify to these cells what their cell fate would be. So now, if you think about evolution and claws that might get longer over the generations, or different sorts of gradual evolutionary changes that we usually picture, this is kind of a ball game changer or a game changer. Because now we can have a single gene that might mutate and sort of really drastically change the whole appearance of this organism, not really in a gradual manner, but in a whole construction of how many legs and how many wings and so forth. So many people are interested in studying how the homeotic genes have evolved and how they're similar or present or not present in different species in the evolutionary tree. Here's another interesting one, and this one you've got to see in lab. It's called antennapedia. And the antenna, part of the antennapedia refers to antenna, and the pedia refers to the feet. And what should be a little branched, kind of articulated antenna structure in this fly has now been converted to legs coming out of the head. And this is really strange, because you would think that if the antenna were going to be changed to something, it might be like another head structure. So why it's a leg, I have no idea. But you can clearly make out little bristles that are characteristic to the legs, some jointed features of fly legs are present in these legs as well. I don't think they could actually use them to walk on because generally they're not really totally fully formed, and then they'd also have to have the correct innervation and so forth. But structurally, that body part is now present coming out of the cockpit head here. So this was work done by a famous classical Drosophilus, and he discovered many of these genes doing genetic screens. And here you can see some of his little milk bottles with cotton stoppers that they used to use to grow fly cultures in. And he eventually got the Nobel Prize for this work. And these genes have been really widely studied, and I think you can see why, because there's these interesting phenotypes. Okay, I'm going to skip this slide for right now and come back to it. And then I want to introduce this slide. So these genes, these 8 genes that can be used, if we look then at the molecular structure of the gene and we say, what's in there? What's special about this gene? These are some of the features. So like any good gene, they're going to have a region that has a DNA coding sequence. So this will start with an ATG, which is your methionine initiation codon, and then you'll have an open reading frame, and that represents the region of the gene that amino acids would be coded based on that DNA sequence. And within that region, which I've represented here as a green box, there is a portion of that coding sequence that we call the homeo box. And it's 180 base pairs long. And if you look from one organism to the next, say various different species of fruit flies, other insects, even other species altogether, and you look at the sequence of that 180 base pairs and how similar it is, it's very, very similar. In other words, it's very highly conserved and has not changed.